Hey guys, I will be making videos on the topic biochemistry from our book First Aid 2023 edition. And let's start with biochemistry, the first chapter. So, in biochemistry, the first topic is chromatin structure. DNA exists in the condensed chromatin form to fit into the nucleus. DNA loops twice around a histone octamer to form nucleosome. Beads on a string and H1 binds to the nucleosome and to the linker DNA, thereby stabilizing the chromatin fiber. DNA has negative charge from phosphate groups. Histones are large and have positive charge from lysine and arginine. In mitosis, DNA condenses to form chromosomes, and DNA and histone synthesis occurs during S phase. Mitochondria have their own DNA, which is circular and does not utilize histones. So the DNA structure is very simple. There is a double helix of the DNA, and we can see in this picture that DNA is wrapped around. Since it is negatively charged, it is wrapped around a nucleosome which is made up of H2A, H2B, H3 and H4. That is 2 times. That is how it makes an octamer. And H1 linker DNA, which we can see, that is attached on the side of that nucleosome. And next we will see the structure of heterochromatin and euchromatin. So heterochromatin is the condensed one which appears darker on electron microscopy. It is labeled H in this picture A. Nu stands for nucleolus. And the darker one, we can see it is heterochromatin. It is sterically inaccessible, thus transcriptionally inactive. So there is increased methylation and decreased acetylation of this one. Heterochromatin is highly condensed. So bar bodies are inactive X chromosomes which can be visible on the periphery of the nucleus. So this question is very much asked in the Usmle, that heterochromatin is the highly condensed one. And in this one, why is it highly condensed one? Because it has increased methylation. So this is very much asked, that what happens due to increase in methylation? The answer is that the chromatin structure becomes very condensed and it becomes inaccessible, as seen in the case of bar bodies. So next is euchromatin, which is less condensed one, and it appears lighter on electron microscopy. It is labeled E in picture A, and it is transcriptionally active and sterically accessible. So EU stands for true, it is truly transcribed, and euchromatin is the one which is expressed. Next is DNA methylation. It changes the expression of the DNA segment without changing the sequence. It is involved with aging, carcinogenesis, genomic imprinting, transposable element repression, and X chromosome inactivation, which is called lionization. So this is also very much asked, what all processes is DNA methylation involved in, which we will study in the further videos, that how DNA methylation it plays a very important role in carcinogenesis. So DNA is methylated in imprinting, and methylation within the gene promoter, that is CpG islands, typically represses or silences the gene transcription. CpG methylation makes DNA mute, and dysregulated DNA methylation is implicated in Fragile X syndrome. DNA methylation usually causes reversible transcriptional suppression, but can also cause activation depending on the location of the methyl groups. Histone methylation mostly makes DNA mute, and lysine and arginine residues of histones can be methylated. Histone acetylation is the removal of histone's positive charge, which leads to relaxed DNA coiling and increased transcription. Thyroid hormone synthesis is altered by acetylation of the thyroid hormone receptor, and DNA histone acetylation makes the DNA active. So this is the most important point, that acetylation makes the DNA active, and it is the removal of the positive charge which leads to this relaxation of the DNA. Histone deacetylation is removal of the acetyl group, tightened DNA coiling, which leads to decreased transcription. Histone deacetylation may be responsible for altered gene expression in Huntington disease. Histone deacetylation deactivates the DNA. Now the structure of nucleotide. Nucleoside is equal to base plus deoxyribose sugar, and nucleotide is base plus deoxyribose sugar plus phosphate, which is linked by 3-5- phosphodiester bond. Purines are made of 2 rings, Ag rings, and pyrimidines are CuT, which is 1 ring. Deamination reactions are cytosine to uracil, adenine to hypoxanthine, guanine to xanthine, and 5-methylcytosine to thymine. Uracil found in the RNA, thymine is found in the DNA, and methylation of the uracil makes the thymine. 5' end of the incoming nucleotide bears the triphosphate, which is the energy source for the bond, and alpha phosphate is the target of 3' hydroxyl attack. And the mnemonic is pure as gold and cut the pyramid. Thymine has a methyl. CG bond, that is 3 hydrogen bonds, are obviously going to be stronger than the 2 AT bonds, which are 2 hydrogen bonds, which means that increased CG content is increased melting temperature of the DNA. CG bonds are like crazy glue, amino acid necessary for purine synthesis, cats purr until they gag. GAG stands for glycine, aspartate, and glutamate. And now they've shown the structure of purine pyrimidine, which is not that important obviously, but remember that pyrimidine contains carboxyl phosphate and aspartate, whereas purine contains aspartate, glycine, and glutamine. Next is the de novo pyrimidine and purine synthesis. Various immunosuppressive, antineoplastic, antibiotic drugs function by interfering with nucleotide synthesis. So pyrimidine synthesis is leflunomide, which inhibits dihydroorate dehydrogenase. 5-fluorouracil and its prodrug capecitabine forms 5-FDUMP, which inhibits thymidylate synthase. DTMP is decreased. Purine synthesis is 6-mercaptopurine and its prodrug azathioprine inhibit the de novo purine synthesis, guanine phosphoribosyl transferase, and azathioprine is metabolized via purine degradation pathway and can lead to immunosuppression when administered with xanthine oxidase inhibitor. Mycophenolate and ribavirin inhibit inosine monophosphate dehydrogenase. Purine and pyrimidines like hydroxyurea inhibit nucleotide reductase, so this is also asked a lot, the mechanism of action of hydroxyurea, which inhibits ribonucleotide reductase. Methotrexate, trimethoprim, and pyrimethamine inhibit dihydrofolate reductase. They decrease deoxythymidine monophosphate, that is decreased DTMP in humans. So methotrexate, bacteria by trimethoprim, and protozoa pyrimethamine. CPS1 mitochondria urea cycle found in the liver, and CPS2 cytosol pyrimidine synthesis, which is found in most of the cells. And now we will also look at the structure, the pathway of purine and pyrimidine base production. It all starts with ribose 5-phosphate, which is converted by PRPP synthetase to PRPP. In the purine synthesis pathway, that PRPP is converted to IMP, and that IMP later on to AMP and GMP. Whereas the pyrimidine base production pathway requires aspartate, glutaminate carbon dioxide by the action of CPS, which is carboxyl phosphate synthetase 2, they are converted to carboxyl phosphate. Later on, orotic acid is converted to UMP with the action of aspartate. Orotic acid is converted to UMP with the action of PRPP, and then UMP is converted to UDP. UDP to CTP or DUDP by ribonucleotide reductase, DUDP to DUMP, and DUMP by thymidylate synthetase to DTMP. Purine salvage deficiencies. So the first of this is adenine deaminase deficiency. ADA is required for the degradation of adenine and deoxyadenosine. So ADA is required for the degradation of adenosine and deoxyadenosine. Decreased ADA leads to increased DATP and decreased ribonucleotide reductase activity. So there is decreased DNA precursors in the cells and decreased lymphocytes. So one of the major causes of autosomal recessive SCID. Next is Leishman syndrome, in which there is defective purine salvage absent HGPRT, which leads to decreased GMP and decreased IMP formation. There is compensatory increase in purine synthesis, which leads to increased PRPP amidotransferase activity and excess uric acid production. It is X-linked recessive. Findings are intellectual disability, self-mutilation, aggression, hyperuricemia, red-orange, SAD, gout, and dystonia and macrocytosis. HGPRT stands for hyperuricemia, gout, pist of aggression, self-mutilation, red-orange crystals in the urine and tense muscles, and treatment is allopurinol and febuxostat. Genetic code features are unambiguous, degenerate, comma-less, non-overlapping, and universal. So unambiguous means that each code specifies only one amino acid. Degenerate means most amino acids are coded by multiple codons, except methionine, which is coded by AUG, and tryptophan UGG, encoded by only one codon. Wobble hypothesis means first two nucleotides of the codon are essential for anticodon recognition, whereas the third nucleotide can differ. Next, there is comma-less non-overlapping, which is read from a fixed starting point as a continuous sequence of bases. Exception is some viruses, and universal means genetic code is conserved throughout the evolution. Exception is there in humans, mitochondria. Next is DNA replication, which occurs in 3' to 5' direction in the continuous and discontinuous Okazaki fragment fashion. It is semi-conservative. It is more complex in eukaryotes than in prokaryotes, but it shares analogous enzymes. So first is origin of replication. Particular consensus sequence in the genome where DNA replication begins may be single in prokaryotes and multiple in eukaryotes. It has 80 rich sequences, that is data box, which is found only in promoters, often upstream, and origin of replication. Next is replication fork, which is Y-shaped region along the DNA template where leading and lagging strands are synthesized. Third is helicase, which unwinds the DNA template at the replication fork. It is deficient in Bloom syndrome, BLM gene mutation. Fourth is single-stranded binding proteins, which prevents the strands from re-annealing or degradation by nucleases. Next is DNA topoisomerase, which creates a single or double-stranded break in the helix to add or remove the supercoils due to unwinding or overbinding of the DNA. In eukaryotes, irinotecan and topotecan inhibit the topoisomerase 1, and adipocyte and adipocyte inhibit topoisomerase 2. In prokaryotes, fluoroquinolones inhibit topoisomerase 2 and topoisomerase 4. Next is primase, which makes DNA primer for DNA polymerase 3 to initiate replication. Then DNA polymerase 3 is found in prokaryotes only. It elongates the leading strand by adding deoxynucleotides to the 3' end. It also elongates the lagging strand until it reaches the primer of the preceding fragment. So DNA polymerase 3 has 5' to 3' synthesis and proofreads with 3' to 5' exonuclease. Drugs blocking the DNA replication often have modified 3' hydroxyl, thereby preventing the addition of the next nucleotide, which is chain termination. DNA polymerase 1 is found in prokaryotes only. It degrades the RNA primer and replaces it with DNA. Same function as DNA polymerase 3 also excises RNA primer with 3' to 5' exonuclease. Then we have DNA ligase, which catalyzes the formation of phosphodiester bond within the strand of double-stranded DNA. It joins the Okazaki fragments, and ligase links the DNA. Next is telomerase, which is found in eukaryotes only, a reverse transcriptase that adds DNA to the 3' end of the chromosomes to avoid the loss of genetic material with every duplication. Unregulated in progenitor cells and often in cancer, downregulated in aging and progeria. Telomerase TAG stands for glory and greatness.